Hello, I'd like to thank you for inviting me to share my research with you regarding soil acidification for improved health of Christmas tree transplants. Here's a roadmap for my presentation. The justification for doing this work is related to trying to find ways in which we can deal with Phytophthora root rot in Christmas tree plantations. It will start out with an experiment I planted in 2010 at a cooperating grower's farm, which we discovered was uniformly infested with Phytophthora root rot. That followed up with an experiment in 2015 in which we acidified the soil to see whether that would protect against Phytophthora infection for newly planted trees, and then end up with follow-up hypotheses to try to explain some of the phenomena that we had experienced with these experiments. But first, a little bit background of Phytophthora root rot biology. Phytophthora root rot is a water mold, which is not a true fungus, that will infect the roots of many species of plants. Certainly Fraser fir is the most susceptible that I know of, and it can affect trees at any size. So we see in this planting trees that were planted probably 2 or 3 years ago to trees that were nearly mature or ready for harvest that have turned brown, died, and dropped their needles. There's one on the right-hand side that shows needles yellowing. That's about ready to die. When you see dead trees, the way that you can diagnose Phytophthora root rot is by taking the knife and scraping off the bark to see whether you find this characteristic cinnamon brown colored discoloration underneath the bark. It's water-soaked appearance, and that vascular tissue has become clogged, leading to the tree wilting and dying. The image of the stem here is from exactly the same tree that I show on the left-hand side here that was experimentally infected with Phytophthora. Now, as a water mold, there are 2 stages of this disease that you need to be especially aware of that are shown here. In the upper left, we have oospores, and that's a stage of the life cycle in which the Phytophthora can exist for many years in the soil, just waiting for conditions to be ripe for the spore then to become active, germinate, and start a new life cycle. So this is one of the main problems with Phytophthora. Once it's present in the soil, it's pretty much a forever problem. The other stage of the life cycle that's important for you to know about and recognize is the activity of zoospores. Here we have an oospore that has germinated to produce what are called sporangia, which are special spore-producing structures that open up and release what are called zoospores. The zoospore is pictured right where I have the pointer. Zoospores have 2 flagellae, which allow them to swim through a film of water in the soil. And so a lot of the biology of Phytophthora is centered around the presence of saturated soil and the ability of the zoospores to move about freely. The zoospores then move to a root where they do what's called encyst, meaning that they lose their flagellae and will settle down and send an infection peg into the root tissue, where then they grow on the interior of the plant to cause the dead tissue that we see. One of the things that you can do to diagnose and isolate Phytophthora is to take a flame-sterilized knife, cut away the tissue, take a tiny bit of that tissue on a sterile knife and insert it through the skin on an apple. And the growth of the Phytophthora within the apple will leave behind the bacteria. And you can go from the edge of the margin of infection within the apple fruit and take a little bit of that material and plate it out onto a sterile media, and you can get a clean culture of Phytophthora to grow. Just as for any disease, we use the disease triangle to figure out why it is that we end up with problems with infections. You need to have a conducive environment, a susceptible plant, and disease inoculum present for disease to take place. The overlap of all three of these conditions can allow disease to occur. So there are a number of different things that you can do to prevent disease, one of which is to modify the environment, which would mean to have a situation in which you don't have standing water frequently. That could mean using raised beds, or to improve the drainage within the entire field by installing drainage tiles. If you have a hard pan, you might be able to use tillage radishes to punch holes through that hard pan and allow water to move through freely. And of course, the focus of my talk will be to change the soil pH, with the idea that the Christmas trees are adapted to growing under very low pH conditions, whereas the Phytophthora might not be able to grow under such extreme pH, low pH, and so that might give the plants an edge. We could also change the host plant to be less susceptible, and that would be to either grow tolerant species, and that will be part of my talk, or you could use the species that are truly resistant, like Momi fir, as resistant rootstock onto which you would graft the tops of something that you really wish to grow. So, for example, I know that John Frampton had used Momi fir and grafted Fraser fir onto the tops of the Momi, because the Momi fir is resistant to Phytophthora root rot. Fungicides haven't really worked particularly well for protection against Phytophthora root rot when talking about field-grown Christmas trees. And I will mention a little bit about inducing systemic acquired resistance pathways. That is a set of tools that triggers the plant's own defenses against infection. Really, the notion about using soil pH really involves changing the environment so that it's no longer conducive to the growth of Phytophthora root rot. The idea is by changing the soil pH to be low enough that the tree is happy to grow, but that Phytophthora cannot grow, we might be able to grow Christmas trees without disease. So that's a basis for these experiments. For the experiment we planted in 2010, we compared the growth of Fraser fir with several varieties. There was Turkish fir, Trojan fir, Nordman fir, and Canaan fir also. And within 3 years of planting in this cooperating growers field, every single Fraser fir had died. You see them here as the brownish red trees in the background of all the green weeds. And what was striking about this is that the Fraser firs were the only trees that died in this experiment. You can see, though it's a little bit hard to see against the background of the weeds, Canaan fir was growing very well. Trojan fir, Nordman fir, and Turkish fir were unaffected, but every single Fraser fir developed root rot and died within 3 years. So the unusual thing about the experimental field is that it was uniformly infested with Phytophthora. Usually when I find Phytophthora root rot in a Christmas tree planting, it's in a low-lying area or perhaps along a swale where water flows and the zoospores can travel right along with the water. And this field was rather unique from my experience in being uniformly infested. So I talked to the grower, and he was amenable for converting it to experimental purposes. And I planted it with Canaan fir, which are tolerant to Phytophthora root rot, and Fraser fir in plots. And so you see for each one plot, and here we have one plot that has no sulfur applied. I have Canaan fir in this row, Fraser fir in this row, and that's paired with another plot in which I had applied sulfur at the rate of 3,000 pounds per acre to drop the pH from the existing condition of 6.2 all the way down to pH of 4. So this is what's called a split plot experiment, and we had 23 replicates. So this group of 14 trees here would be one full replicate, and there were 23 replicates in this field. The numbers within these little blocks indicate that different types of root dip treatments, including potassium phosphate, which was included to explore whether there'd be effects from the systemic acquired resistance pathway being induced. Through this experiment, I used a rating system to indicate the health of the root systems without having to dig up the trees and actually look at the roots. So a 0 rating was a dead tree. 1 would be a bright yellow tree. 2 would be a greenish yellow. 3 is a yellowish green. And 4 would be a completely healthy plant. You can imagine my surprise when even at the end of the first year of having set up this experiment and having planted in the trees, which was in 2015, we saw a tremendous benefit from having incorporated the sulfur based upon the overall appearance of the trees. To orient yourself to this graph, the species here are Canaan on the left, Fraser on the right. So these 4 bars are in order: no sulfur and no prophyte or potassium phosphate, potassium phosphate, no sulfur, sulfur with no prophyte, and sulfur with prophyte. And then those treatment combinations are the same here with the Fraser fir as well. Basically color-coded. The green is a combination of blue and yellow, the yellow meaning sulfur, and the dark blue meaning that the prophyte was included. So from this graph you can see a couple things that are very interesting. Only Fraser fir benefited, and it benefited quite a lot, by including prophyte, which triggered the systemic acquired resistance pathway, and that was statistically significant, indicated by the single asterisk here. Canaan fir probably did not benefit from the addition of prophyte, possibly, and I'm speculating here, because as having innate degree of resistance to Phytophthora to begin with, it may constitutively or always have turned on those defenses against infection by Phytophthora. So it's only in the susceptible plant that you see the benefit from turning on the defenses with prophyte. The sulfur, these green bars compared to the blue bars, showed a benefit in the improvement of the color rating for both Canaan fir and Fraser fir. In the second year of growth, there wasn't much of any differences with the overall color of the plants. However, there was with the overall growth of the leaders. This graph shows the comparison of the plants in which there was no sulfur applied in the plots versus those with sulfur with respect to the current season growth of the leader in centimeters. And you can ignore the colors and the number of bars that are involved here. There are a number of treatments, but if you can consider the average from this group where there was no sulfur, and then compare that with where sulfur had been applied, there was a doubling in the amount of terminal growth where sulfur had been applied. This slide shows the effect on the tree mortality from having incorporated sulfur, and it compares Fraser fir versus Canaan fir. So Canaan fir overall had much lower mortality than the Fraser fir. And you can see for both species that sulfur incorporation improved the survival, reduced the amount of cumulative mortality. And I have to admit that this is only part of the story, though, because we had a drought for the first 3 years of the experiment, and then in 2018 we had 62 inches of precipitation, which was about 20 more inches of precipitation than normal for Connecticut. And at the end of that year, and beginning of 2019, the actual Fraser fir mortality rose close to 100%. So that was 4 years after planting. So certainly we had conditions in these first 3 years that were dry enough that we didn't see such extreme mortality. And that leads to the overall advice I can give you. If you have a field that's heavily infested with Phytophthora, you certainly would not want to plant a species as highly susceptible as Fraser fir, or you would be losing them continually from root disease whenever you had weather conditions conducive for disease development. So my advice would be in this highly infested field only to plant Canaan fir or some other Phytophthora tolerant species, and to also acidify the soil to some degree. But this leaves a couple of questions unanswered. First of all, why was it that the growth was improved in these trees? And why didn't the trees survive the wet year? And to look at this in more detail and try to tease it apart, I'm going to try to apply the process of strong inference, which means that I consider all plausible explanations or hypotheses for why we saw the benefit and improved health of the trees, and then systematically test or try to falsify each of these hypotheses. And that this is an understanding that the various possible explanations don't have to be mutually exclusive. If I find evidence supporting one of these ideas, that doesn't mean that the other ideas are necessarily false. And these are the possible mechanisms that I've come up with for how sulfur improved the health of the Christmas trees. The first hypothesis is that simply Phytophthora is intolerant of low pH. That was the original idea behind setting up this whole experiment. The second hypothesis is that elemental sulfur might act directly to kill the Phytophthora organism. Elemental sulfur is sometimes used as a fungicide to control things like powdery mildew, so that certainly stands as a possibility. Third hypothesis is that the conversion of sulfur by microorganisms to sulfuric acid liberates hydrogen ions, which as they leach through the soil will then liberate calcium ions into soil solution. And it's already known that high concentrations of calcium ions can interfere with the biology of Phytophthora zoospores. The fourth hypothesis is that reduction in the pH would make manganese highly available to the plants, and higher manganese concentration within the tissue of the plant might allow the plant to ward off disease. And then the fifth and sixth hypotheses are somewhat related, but they're flip sides to the same coin, in that lowered pH might favor bacteria or fungi that are antagonistic to Phytophthora, and higher pH might favor organisms that make Phytophthora infections worse. So some of these hypotheses can be directly tested in the laboratory. For example, a direct impact of pH can be tested in the laboratory by manipulating the pH in growth media and seeing how that affects the growth, survival, and the ability of Phytophthora to cause infections. And this graph is rather busy, but it shows the results of an experiment testing the pH hypothesis. And in this graph, I show 4 different species of Phytophthora that have been isolated from Christmas trees, diseased Christmas trees in Connecticut. So there's Phytophthora cactorum, Phytophthora calmania, Phytophthora pini, and Phytophthora plurivora. And we set up growth media, which we then inoculated in the center of a petri dish with Phytophthora, and then measured the radial growth over time. And then we compared the growth at these various pHs with the pH at 5.8, which was maximal growth. And we get these really clear dose-response relationships. So I can identify the pH at which we'll have a 50% reduction in the amount of Phytophthora growth. So for example, for Phytophthora cactorum, that has a pH of 4.3 will reduce the radial growth 50% compared to the higher pH. And all of these Phytophthora are sensitive to the pH conditions in which they're growing. However, I must say that this is somewhat of an artificial test because this is a growth of mycelium for Phytophthora, and that is growing typically within the tissues of the plant. And in that protected environment, they would never be exposed to pH of this low. A more relevant question will be to do the same kind of bioassay, but with Phytophthora zoospores, which are exposed to the acid environments outside of the soil, outside of the root in soil solution. So for our first hypothesis, we do have evidence that radial growth of Phytophthora mycelium is affected by pH, but that's probably not really ultimately what's important. We need to test the ability of zoospores to affect bait in an acid environment to know whether this is likely to have directly caused the effects we saw. So the second hypothesis is related to the release of calcium into high concentrations in soil solution. And we can test this by changing the calcium ion concentration and then observe growth, survival, and infectivity of various stages of Phytophthora. When we look at the effect of calcium concentration on the radial growth of Phytophthora mycelium, basically the higher the concentration we had, there was very little, if any, effect on the radial growth. So it's practically flat here. And so what that means is that certainly with respect to the mycelium of Phytophthora, the calcium concentration really is of no significance. As I alluded to earlier, it already has been demonstrated by other researchers that a high concentration of calcium in the soil solution, or in this case in nutrient irrigation solutions for hydroponically grown crops, can interfere dramatically with the biology of Phytophthora zoospores. So in this published report, you see in the water control that you have a lot of mobile zoospores. 90% of the zoospores are mobile. And as you increase the concentration of calcium to 10 millimolars, or milliequivalent, and then up to 20 milliequivalent, that the percentage of mobile spores drops down dramatically to the point where at 30 milliequivalents of calcium, 30 millimolar concentration, there is no activity of Phytophthora zoospores. So this is something I'm currently investigating to see whether we can reach that extreme level of calcium ion concentration by liberating the calcium from exchange sites in the soil through soil acidification. One of the reasons why I think that this is plausible is that when we took the soil samples from the non-acidified plots and the plots to which we had applied sulfur in 2020 and evaluated those soil tests, those soil samples with soil tests, what we found was that for the non-acidified soil, the calcium concentration in the soil extracted with the standard soil test method was around 2,000 parts per million, and that had dropped to about 620 parts per million where we had acidified the soil with sulfur. That means that over the course of about 5 years, we had leaching of 1,378 parts per million in the soil, and that would convert in a 6-inch soil profile to about 2,756 pounds per acre. Well, a curious thing about this too is when you look at calcium versus magnesium and potassium, all of the base cations that might be liberated from the cation exchange sites in the soil, it appears that calcium in particular has a much larger proportion of loss to what was present in the soil than these other minerals. There's a much smaller percentage change when you go from 88 to 49 or 94 to 88 than there is from 2,000 to 620. So now the real question is, we can see direct evidence that we lost calcium from the soil profile through leaching. And that meant that the calcium was liberated from cation exchange sites into soil solution, but we don't know whether the concentration in the soil solution ever would have reached the 20 to 30 millimolar calcium ion concentration required to shut down the biology of the Phytophthora zoospores. So this is something that I am investigating right now to find out just what those concentrations can be in the soil solution when we initially acidify the soil. To test the sulfur hypothesis is rather difficult because sulfur is not water soluble. And so what I do is I make a suspension of an agricultural flowable sulfur product that's used for control of, let's say, powdery mildew as a foliar spray. I make a suspension of that flowable sulfur product with the auger and then see how the Phytophthora grows. And as with the calcium experiment with the radial growth of the various Phytophthora species, there doesn't seem to be much of an effect of the sulfur. It is statistically significant, but probably not a biologically meaningful effect from including sulfur in the media. To test the hypothesis that we change the mineral content of the plant tissues through soil acidification, and specifically with an interest in manganese because that's been related to plant defense against diseases. What I did is that I took samples in 2020, I dried them and ground up the needles and then sent them out for analysis of their mineral content. You can see the relative concentration in this graph. It's on a log scale, so they all fit onto one graph, of boron, calcium, copper, iron, potassium, magnesium, manganese, phosphorus, sulfur, and zinc, with the plant tissues that originated from the non-acidified plots, those are the clear bars, versus those plots that had sulfur incorporation. Those are the yellow bars. When you look at the log scale, it really makes it clear that there's no really major difference. There might be statistically significant differences here in calcium, copper, and zinc, and iron, but the only mineral that was really changed dramatically was the manganese concentration within the plant tissues. With soil acidification, we boosted the amount of manganese taken up by those plants. So the tissue concentrations went up from 18 to 250 parts per million on average. So overall, I can say that the manganese hypothesis is supported. And that might have a lot to do with the improved color of the plants after having acidified the soil. Acidification through the addition of elemental sulfur causes the sulfur to be changed into sulfuric acid as those protons, or hydrogen ions, are moving through the soil. They are kicking other cations off of exchange sites into the soil solution and making them really hyper available to these newly transplanted trees. And so part of the reason why we see much darker green foliage can be that their roots are being bathed in a much higher concentration of these micronutrients. Well, what about the microbial interaction hypotheses? That's not something I would be able to tackle on my own, so I have collaborated with scientists from the Connecticut Agricultural Experiment Station. Dr. Blair Stephen is a microbial ecologist and one of the world experts on soil microbiomes. So what we did is that we teamed up with support from the Christmas Tree Promotion Board funding, and we did a comparison of the soil rhizosphere microbiome comparing the roots from plants that had grown under very low pH with the higher pH. Now, I'm not going to claim that I can really fully explain this graph. This is one that Blair put together from our data, but it is quite striking that the microbes that you find around the root system in the acidic environment have very little similarity to the microbes that you find in the non-acidified soil environment. What this graph shows is that these green dots are for the non-acidified plots. Each dot is a species of bacterium that was found in the root system. And the red dots are showing individual species that were found in the soil that had been acidified, and there's very little overlap. The dots that are gray are the ones that are in common, and there are probably about 2,000 to 5,000 species here that really aren't in common amongst the root systems, surrounding the root systems in these two different pH conditions. These can be looked at in a different way, and that is, it's like principal component analysis or cluster analysis. And you can look at the entire community of microorganisms around the root systems. For the non-acidified plots, the fact that these points, each point representing a single tree that we had sampled, each of these points are very tightly clustered with the other points from the similar environment, meaning that the communities all are very similar around the root systems of those plants with a higher soil pH. When you look at the soil microbial community around the root systems for the plants that had experienced the lower pH conditions, each one of these spots is really far apart from all the other red spots, meaning that there is a unique microbial community around the root systems for every one of those trees, which is rather a shocking thing to consider, that we shook things up by having added the acidity to the soil, and it never has coalesced into, let's say, a whole group of little spots all off in one area here. Rather, each one of these root systems has its own complement of bacteria. There's another side to this that's interesting, and that is that we can take the bacteria from around the root systems and explore to see whether we can find bacteria that are really antagonistic to the growth of Phytophthora root rot. And here I show a petri dish in which I've taken one isolate of bacteria. It's a pure isolate. And I've inoculated it either with 2, 4, or 8 little spots surrounding a location where I introduced a chunk of agar that had Phytophthora growing on it. You see in these spots, 2, 4, and 8, there is no growth of Phytophthora visible off of where I introduced the colony. Whereas with the 0 or untreated check, the Phytophthora grew quite nicely. You'll see that it didn't grow equally around that chunk of agar and out onto the petri dish. It's showing the effect, it's called a zone of inhibition or antibiotic effects. Exuded by these bacteria, there are compounds that are antagonistic to Phytophthora and are protecting or keeping the Phytophthora from growing anywhere close to those bacterial colonies. Now this is a really interesting phenomenon and one that I think that could be turned into something useful. Currently I am exploring the ability to take these individual isolates of bacteria and see whether we can inoculate roots to protect them from Phytophthora infection. However, the curious thing about this is that cultural bacteria that suppress Phytophthora are remarkably common both in the acid and the non-acidified soil conditions. So the fact that the bacterial communities look very different in the acid versus the less acid soil may not have any bearing on the overall health of the Christmas trees that we have planted. And that bears keeping in mind because when we finally did have that major rain year in 2018, the susceptible trees succumbed to Phytophthora root rot. One of the challenges facing us if we wish to make use of bacteria to inoculate the roots to prevent infection with Phytophthora is that these involve very species-specific interactions. On the left in this column, you see designations of various isolates of bacteria we took from the soil. And then the larger the number you see here, the greater the degree of suppression there was in the growth of different isolates of Phytophthora. This is all in petri dishes. And so we could almost have a unique combination of interactions for each species of bacteria. There are unique combinations or patterns of which species of Phytophthora are inhibited from growing. And so if we wish to make practical use of these bacteria, we would need to have a mixture of a number of them to be able to to theoretically prevent the growth of any of the Phytophthora that could be infecting our Christmas trees. So in summary, looking at our original list of possible hypotheses to explain why the trees look so much healthier, we do have evidence. The red check marks indicate that we have positive evidence supporting the hypotheses. We know that Phytophthora is intolerant of low pH, but that's based on the mycelial growth. We still need to test that with the zoospores. It does not look as though elemental sulfur acts as a biocide, at least so far. And that's very unlikely to be the explanation, because sulfur has very, very low water solubility and probably can't be present in the soil solution as elemental sulfur. It really has to be converted to sulfate, and it would be the sulfuric acid rather than the sulfur itself and the sulfate that would be likely to interact with the Phytophthora biology. The calcium hypothesis is certainly supported from the point of view of our being able to document that we lost calcium from the soil into soil solution. I just need now to find out whether we could reach the type of concentration that would be necessary to interfere with the zoospore biology. The fourth hypothesis related to manganese concentration in tissues was supported. And the hypothesis that lowered pH favors antagonistic microflora certainly might be supported, even though we found antagonistic bacteria in both lower and higher soil pH conditions. The one that I showed in the Petri dish that has that bluish color, we only recovered from where there was low soil pH. The last hypothesis is one I haven't yet figured out how I might be able to examine, that the higher pH could favor the organisms that make Phytophthora infections worse. That would be a very difficult thing to try to tease apart. So in summary, I would say the first piece of advice is don't plant susceptible species in heavy soils where disease is already present. Even though soil acidification slowed down the progress of disease in the susceptible Fraser firs, we still lost all of the susceptible Fraser firs in that field. And the fact that it takes a few more years for them to die just means that you have the heartache of pulling up red-colored Christmas trees that are a little bit taller and took a little bit longer to die than if you had not acidified the soil. Soil acidification benefits young trees through making micronutrients super available. And so initially we see that the trees are much better color, they have better growth performance, but this is at a cost of losing a lot of calcium from the soil. And so I think that rather than targeting an extreme pH like a pH of 4, targeting a pH between 5 and 5.5 is much more reasonable. And it means that we won't end up then with mineral deficiencies of calcium in particular. One of the things that I'm exploring right now is to see whether we could maintain calcium divalent cations at a very high concentration in the soil. This, from other researchers' work, would suggest that we could stop the ability of zoospores to move freely through soil solution to infect roots. And If we're able to set up a situation where there's a constant flux of calcium flowing through the soil solution at a high enough concentration, that theoretically at least would be able to protect sensitive species from Phytophthora infection. So this is something that I'm continuing to investigate. Thank you very much.